First, I'm excuse me, John Slattery, the Vice Dean for Research and Graduate Education in the School of Medicine. And it's my pleasure on behalf of the Council on Research and Graduate Education, which is an elected council of faculty who chooses the speakers for the SIM series, to welcome David Veesler today. David took his master's in structural biology from Aix-Marseille University in 2006 and completed his PhD also in structural biology from the same university in 2010. And he went on right after that to go to the University of Zurich as a visiting researcher, and then hopped across the pond in 2011 to do a postdoc at Scripps Research Institute. In 2015, he joined our Department of Biochemistry as an assistant professor. David has, I think as all of you know, has gotten off to a truly outstanding start. To name just a few awards and recognitions, in 2017, he was selected as a Pew Scholar in the Biomedical Sciences. 2018, Burroughs Wellcome Fund investigator. And in 2022, very important recognitions, an Amgen Young Investigator Award, but then also the coveted NIH Director's Pioneer Award, which is truly outstanding recognition. As you all know, David's work has been in the area of structural biology, the determination of structures employing cryo-electron microscopic techniques. The story he's going to be telling today looks at the structural biology of the SARS-CoV-2 virus. A few weeks after the SARS-CoV-2 isolate was sequenced, and that sequence was published, David's lab demonstrated that ACE2 is a functional receptor for the virus, and published atomic level structural detail of the viral spike protein, and that therefore the infection machinery. So he was one of, I think, two laboratories providing the blueprint for all the work that's been done to design therapeutics and vaccines. His work is continued. He'll be telling us that story today, with the culmination in the form of a vaccine that's actually in clinical trials right now. His talk today is entitled Structure-Guided Design of SARS-CoV-2 Subunit Vaccines. David, take it away.
Thank you very much, John, for the kind introduction and for the invitation. So I'm really happy to be here today. And so as you mentioned, my lab is interested in infectious diseases. We study how pathogens enter host cells and how the immune system responds to infection. And so we had been working on coronaviruses for many years before the pandemic started. And today I will be telling you about the work we have been doing on coronaviruses, especially since January last year. But just to give some historical context, coronaviruses are believed to have been infecting humans for several centuries, but they were not really into the focus of much attention. Things changed quite a bit at the beginning of the 21st century with the emergence of SARS-CoV in 22 in southern China, which rapidly spread to 5 continents, leading to 9,000 infections and about 10% fatality rates. So this was the first large-scale epidemic of the 21st century, and SARS-CoV is believed to have originated in bats and was transmitted either directly to humans or through palm civets, and it is possible that both mechanisms of transmission were at play. In 2004, SARS-CoV disappeared, although we did not have vaccines or therapeutics, and never came back. However, in 2012, MERS-CoV emerged in Saudi Arabia and is still circulating there. It has almost become endemic. MERS-CoV has a 35% fatality rate, which is a lot higher than SARS-CoV or SARS-CoV-2. And although MERS-CoV is believed to have originated in bats, transmission to humans directly happens from camels that are also infected. So what did we know in between 2012 and 2020? Were there any evidence that more coronavirus emergence could occur? So actually a lot of work had been done at the interface between wildlife and humans, and a bat virus highly similar to SARS-CoV that is believed to be putatively the progenitor of SARS-CoV was discovered in 2013 in bats in southern China. And many other reports followed to explain that many coronaviruses are circulating in bats, some closely related to SARS-CoV, some closely related to MERS-CoV. And the last piece of evidence was the publication a few years ago showing that people living close to caves in southern China where SARS-CoV-related viruses were found in bats, some of them had actually probably been infected by related viruses because analysis of their serum antibodies showed cross-reactivity to these SARS-CoV-like viruses, suggesting undetected cross-species transmission occurring in these regions. So what are coronaviruses looking like? As you mentioned, John, I'm a structural biologist. I love visual science. So let's start by looking at the whole virus. And this electron micrograph shows how coronaviruses are shaped. And I would just like to draw to your attention on the fact that these viruses are decorated by a prominent protein called the spike glycoprotein. It forms homotrimers, as shown here, encoded in the viral membrane. And this protein is in charge of attachment to the host cell surface receptor, and then fusion of the viral and host membranes to allow initiation of infection. So before diving into our SARS-CoV-2 work, I just want to show you a movie that is really culminating from 5-6 years of coronavirus work in my lab that explains how we're thinking of how the spike glycoprotein works. So it's a movie performed in collaboration with Janet Iwasa at the University of Utah, and that was based on MERS-CoV spike, but everything I will be showing actually holds true for SARS-CoV-2. So viruses are decorated by these homotrimers of spike glycoproteins. Each protomer has a distinct color, and the receptor binding domains are the parts that you can see opening and closing in each protomer. I will be calling receptor binding domains RBD moving forward. So when the RBDs open up, they can interact with the host receptor, and host proteases can cleave the spike glycoprotein to activate it to lead to membrane fusion. So the host membrane at the top and the viral membrane at the bottom are merged together, allowing viral genome delivery and infection to start. So SARS-CoV-2 emerged at the end of 2019 in the region of the Wuhan city in China. And at the end of December, I could see on my social media feed some reports of doctors describing symptoms similar to SARS among hospitalized patients. So we keep a very close look on these initial reports. And on January 10, 2020, the sequence of the first SARS-CoV-2 isolate was released. And it showed that SARS-CoV-2 most likely originated from bats as well, due to high sequence similarity of the genome and key proteins to related viruses sequenced from bats. I'm sure many of you have heard about Malayan pangolins, on which 2 distinct SARS-CoV-2 isolate sequences were found. And at one point, they were suspected to have participated in transmission, but right now, it seems very unlikely that pangolin contributed in any way to transmission to humans. So SARS-CoV-2 has really spread like wildfire, leading to the current COVID-19 pandemic with over 114 million cases and more than 2.5 million fatalities. And this is the reason why we are having this lecture on Zoom today. So as I mentioned, on January 10th, the sequence of the first isolate was released, and we focused on designing constructs that very day to be able to order them as fast as possible and contribute to understanding this newly emerged virus. So Lexi Walls in my lab actually developed one of the very first pseudovirus entry assay system in the world. And in a nutshell, it consists in using a non-replicating virus backbone in which we graft the glycoprotein of interest, in that case SARS-CoV-2, which allows you to investigate spike-mediated entry at a low biosafety level. What Lexi was able to show a few weeks after the first sequence was released is that ACE2 is an entry receptor for SARS-CoV-2. It's actually necessary and sufficient for entry into the HK cells. And the reason we suspected ACE2 could be a receptor is due to the high sequence similarity between SARS-CoV and SARS-CoV-2. To understand how SARS-CoV-2 enters host cells, Lexi worked closely with Youngjun Park to determine structures of the spike glycoprotein using CryoEM. And I'm showing you on the left-hand side, one out of the two structures we determined from a single data set, showing the RBDs, the receptor-binding domains, in a conformation that we call closed, because they are not competent to interact with the receptor ACE2. However, we know this protein changes conformation, and from the same data set, we were able to actually tease out a second conformation in which one of the RBD is in the open state, and that protomer can interact with ACE2, as is shown here. So the SARS-CoV-2 spike glycoprotein, which is really the infection machinery, is highly similar at the protein level to the SARS-CoV spike glycoprotein. And this comparison holds true when we looked at the end-link glycosylation pattern of this protein. It is a heavily glycosylated protein with 66 glycans decorating the trimer, 22 of them on each protomer, and 20 out of 22 glycans are conserved between SARS-CoV and SARS-CoV-2. What it tells us is that the antigenic landscape that will be recognized by antibodies is actually relatively similar between these two viruses, and that's important for what I will be showing you on the next slide. So as John mentioned in the introduction, these structures have really provided a blueprint to interpret the effect of mutations that are arising in variants and to guide the design of vaccines and therapeutics. And these structures have literally been used by thousands of groups worldwide since we released them. If we analyze the sequence conservation with this subfamily of viruses, which we call sarbecovirus subgenus, so SARS-CoV and SARS-CoV-2 are both members of the sarbecovirus subgenus, we can see that the sequence conservation is quite different if we consider the apex, which we call the S1 subunit, which is fully conserved, or the fusion machinery of the stem, that is also called the S2 subunit, which is highly conserved. We interpret these differences as resulting from the necessity to interact with different receptors for some coronaviruses, although I showed that SARS-CoV and SARS-CoV-2 interact with the same receptor. And the fusion machinery, which really is the business part as far as entry is concerned, needs to be more conserved to be functionally efficient. So based on this sequence conservation, we hypothesized that if one was able to identify antibodies cross-reacting between SARS-CoV-2 and SARS-CoV, they would most likely be binding to the fusion machinery. And Lexi had actually been working in collaboration with Andy Maguire at the Fred Hutch to immunize mice with the SARS-CoV spike glycoprotein a couple of years ago, and show that the hyperimmune plasma were able to neutralize spike-mediated entry of SARS-CoV, as shown here for these 4 animals, and compared to the control group. The question we asked is it possible for SARS-CoV elicited antibodies to neutralize SARS-CoV-2 spike-mediated entry? And the answer is yes, and that really provided proof of principle that cross-reactivity is possible between these two coronaviruses that spilled over in humans. So my group had been working for many years in collaboration with David Veesler at Vir Biotech, and we had actually characterized the first human neutralizing antibodies in complex with spike glycoproteins for MERS-CoV, which I will not be showing, and for SARS-CoV. That's the work that was done by Alex Cheung, a former postdoc in the lab. The antibody we found was very potent against SARS-CoV, binding to the RBD and preventing attachment to the ACE2 receptor. But unfortunately, this antibody did not cross-react with SARS-CoV-2. However, in collaboration with David's group, we undertook a very large screening effort from serum samples obtained from the very same individual, a SARS survivor from a 2003 infection, to try to identify cross-reactive antibodies. And this very large screening effort led to the identification of a handful of antibodies that cross-reacted efficiently with both spike glycoproteins of SARS-CoV-2 and SARS-CoV. One of them, shown in red, which we call S309, was actually neutralizing spike-mediated entry for both SARS-CoV-2, SARS-CoV, but also a bat-related virus called WIV1. In collaboration with Mike Diamond, we showed that the antibody S309, as shown in red again, neutralizes SARS-CoV-2 authentic viral entry into host cells and could even synergize with a weakly neutralizing antibody isolated from the same survivor, showing an increase in neutralization potency when you combine the two antibodies. So to understand how S309 could cross-react with the two viruses, Young determined structures of the spike glycoprotein complex with this antibody. And here again, we identified the same conformational states than the ones we had identified previously for the spike in the absence of any antibodies. The color scheme is the same. Each protomer has a different color, and the antibody is shown as two shades of magenta and purple. On the left-hand side, we can see that here we have one RBD open, and the other two are closed. And Young obtained a second structure with the 3 RBDs closed, showing that the antibody could recognize the spike glycoprotein independently of its conformational states. Zooming up, we realize that the antibody not only forms contact with the protein part as expected, but also forms intricate contact with the glycan at position 343, which accounts for about 25% of the surface buried at the interface between the antibody and the viral targets. These glycan, as well as the residues recognized by the antibody, are very conserved within the sarbecovirus subgenus, explaining why this antibody not only cross-reacts, but also broadly neutralizes SARS-CoV-2 and SARS-CoV. So to understand how the antibody works, we wondered if it could block attachment to the host cell receptor, but the footprint of the antibody as shown here is actually distinct from the ACE2 footprint. The antibody binds on the other side of the RBD. And in solution, we actually cannot see competition for binding between ACE2 and the antibody to the SARS-CoV-2 RBD or the SARS-CoV RBD. So we are still trying to understand and uncover the mechanism of action of this neutralizing antibody. What's interesting is that S309 can trigger antibody-mediated cell cytotoxicity, as shown here in red. It's actually doing pretty well as antibody-mediated cell phagocytosis. So these effector functions are expected to provide additional protective effects in vivo. And S309 entered a phase 3 clinical trial last year in the US, and we're actually expecting in the coming weeks the results of this clinical trial. So this work was done very early before SARS-CoV-2 infected individuals were found in large quantities in the US or even in Europe. But that changed very quickly. And so one of the questions we set out to answer is, what is the nature of antibody responses in SARS-CoV-2 infected individuals? Because the work I just showed you, again, resulted from samples obtained from a SARS-CoV infected individual. So in collaboration with David Veesler's group, we started a very large serological and structural study. From 650 individuals, we looked at antibody responses against the spike glycoprotein as shown here, as well as the distinct domains comprising the spike and the nucleoprotein. What's important is that the data is rendered to show that hospitalized individuals in red, and what we can see is that antibody titers are much, much higher in hospitalized individuals, no matter in which fraction you are looking at, as compared to non-hospitalized individuals. We interpreted that as resulting from higher viral titers in hospitalized individuals, leading to production of higher titers of antibodies. What's interesting is to see that the RBD is actually immunodominant. Most antibodies target that region of the spike glycoprotein. And we also saw that antibodies capable of blocking ACE2 binding were actually present at high titers in most 3/4 of infected individuals that were hospitalized, as compared to much lower titer and much lower fractions, 20 and 10% in symptomatic or asymptomatic non-hospitalized individuals. And the importance of such antibodies is demonstrated by this plot that shows direct correlation between the ability for antibodies to block ACE2 attachment and SARS-CoV-2 neutralization potency. Actually, this ACE2 blockage of binding has been implemented in some commercial kits as a surrogate for the presence of neutralizing antibodies that does not require a cell-based assay. So because the RBD is immunodominant, we ask, what is the contribution of the RBD to neutralizing activity? So we depleted serum samples from infected individuals from the antibodies targeting the RBD, and you see on the left before and after, on the right depletion. And what we saw is that neutralizing activity was decreased by about tenfold after doing that. So that tells us that the RBD is not only immunodominant, but it is also the main target of neutralizing antibodies, accounting for most neutralizing activity present in serum of infected individuals. So Young undertook a very large effort to characterize the fine specificity of epitopes targeted by these antibodies to correlate serology and structural studies. And what Young found is that the receptor binding motif, which is the target of, which is the region bound by ACE2, is targeted by at least 2 classes of antibodies recognizing distinct antigenic sites. These 2 antibodies are shown here in orange and cyan or teal. And the main difference between these 2 sites, beside the different angles of approach, is that S2H14-like antibodies, as shown in teal, can only recognize the spike when the RBD is open, as is the case for the natural receptor ACE2. However, the orange class of antibodies, S2H13-like, can bind both the open and closed conformational states. Both of them are capable of blocking ACE2 binding by overlapping with its recognition sites, as we can see based on the structure and in solution, and that holds true for S2H13-like antibodies as well. So this is a very, very important site because, as I just showed, ACE2 blocking correlates with neutralization potency. Then Young and Lexi worked along with Ale Tortorici to characterize a cryptic antigenic site that is here on the side of the RBD, and that can be accessed by antibodies only when 2 neighboring RBDs are open. Here you can see 2 neighboring RBDs, the third one is here, and the antibodies can really not get in due to steric hindrance if 2 RBDs are not open. They determine 3 structures of the 3 antibodies shown here in red, yellow, and purple. And as you can see, they differ by the distance from this region of the RBD, which is the ACE2 attachment site. Because these antibodies conformationally select for the spike in its open state with multiple RBDs open, it actually shifts the equilibrium and lead to triggering of the fusion machinery as observed in CryoEM by uncoupling of the S1 subunit trimer, which is the apex of the spike shown here, from the fusion machinery, which would be here and that has disappeared. So the fusion machinery, although still covalently linked to the S1 trimer, has been triggered and is disordered to such a point that we cannot even visualize it anymore. So these antibodies actually act by mimicking a bit the effect of the receptor by triggering the refolding events leading to membrane fusion. We don't really know how that contributes to neutralization, but one could envision that adding these antibodies to purified viruses will lead to premature triggering and inactivation of the virus. But again, we know how it works in vitro. We do not know how this mechanism plays out in vivo. So what's interesting is that the neutralization potency of this family of antibodies inversely correlates with the distance from the ACE2 binding site. S304 binds away from the ACE2 binding site and does not overlap with it. Perhaps the glycans from ACE2 will be sterically hindered by the antibody, and this antibody is actually a weak neutralizer. S2A4 does not share the same binding site, but the antibody Fab constant domains are expected to clash with ACE2, and this antibody actually neutralizes slightly better. S2X35, however, is binding at an epitope that overlaps the ACE2 binding site. And this antibody is a much, much better neutralizer, suggesting that although it's a distinct antigenic site, here ACE2 blocking of ACE2 attachment is again the main mechanism for neutralizing the virus. So this work, which I summarized in 2 slides, really led us to defining an antigenic map of the SARS-CoV-2 RBD, which is a target of most neutralizing activity in convalescent individuals. And using these different antibodies, we were able to ask in a quantitative way the proportion of antibodies targeting each antigenic sites in hospitalized or non-hospitalized individuals. And the bottom line is that the site which we call site 1, which is the ACE2 attachment site, is the most prevalent in terms of targeting by antibodies. And that's a good news because, again, the binding of antibodies blocking ACE2 correlates with neutralization potency. And it seems like across the board, this is the site that is immunodominant, which we know, based on what we have shown, is comprised of multiple antigenic sites. So this is a very large effort that was done in collaboration between our group and via biotechnology. And out of 800 monoclonal antibodies characterized, 2 of them really stood out due to their ultra high neutralization potency. I'm showing you here neutralization curves for the 2 antibodies. It's a dose response curve. The more antibody, the more neutralization. But the midpoint is in the single-digit nanogram per ml, which is really, really high. These are the most potent antibodies found to date. And we actually showed that these data hold true in 4 different neutralization assays to be as confident as possible. To add to the unusual characteristics of these antibodies, we saw that binding was affected by pH in such a way that if we drop the pH, which we know leads to closure of the RBD toward the closed spike, one of them, S2E12, will exhibit decreased binding to the spike, whereas the other one, S2M11, will show increased binding to the spike. And by doing a range of binding assays that I will not describe in the interest of time, we knew that it was not just a pH-dependent effect on protonation or deprotonation of our spike, but that was actually due to a quaternary effect. So Ali took the lead in characterizing structurally these antibodies to understand how to explain these biochemical properties. And the first of them, which is called S2E12, was actually shown throughout this work to bind to the RBD at the receptor binding motif shown here. And we have a zoom-in view on the right, which completely overlaps the ACE2 binding site. However, S2E12 is especially interesting because not only we block ACE2 binding, but it is binding in such a way that it remains away from the mutations that have been recently identified in the variants of constant that have emerged. So S2E12 binds just as well to the B117, as known as UK variant, and will not be affected in that regard. The second antibody, S2M11, was very surprising because the structure showed that it binds across 2 neighboring protomers. If we zoom in, we can see that CDRH3 is bridging 2 neighboring RBDs, and we have contacts with the same glycan as the one that was contacted by S309. And S2M11 not only overlaps with the ACE2 binding site, but really locks the spike in the closed state, which will even prevent exposure of the ACE2 binding site. So here we have really a dual effect, blocking of receptor attachment, but also preventing the conformational changes leading to membrane fusion. So I showed you a range of different binding modes, and I explained that some antibodies were more potent neutralizers than others. But at the end of the day, the most important is, are they protective in vivo? So we decided to ask the question using a golden seal and a hamster model of SARS-CoV-2 infection using these 2 ultra-potent antibodies. And this work was done in collaboration with Johan Nates at KU Leuven. And what the work showed is that very low quantities of antibodies, 1 mg per kg, either individual antibodies or cocktails of them, decrease significantly viral genome RNAs in the lung of infected hamsters. And more importantly, here we are looking at viral replication. We see that viral replication is completely shut down by all antibody regimens that we administered in a prophylactic way. So that's very encouraging because, as you know, many antibodies have either been evaluated in clinical trials or have already received emergency use authorization. And we see that potent neutralizing antibodies have a very good effect in hamster, and that has been shown by now in clinical trials. They are also helping in the clinic. So at this point, I hope I convinced you that the RBD is really, really important, but it's not the only target of neutralizing antibodies. And recent work has emerged from our group and others that the N-terminal domain, or NTD, is also a target of potent neutralizing antibodies. So in collaboration with David's group, we looked at the specificity of antibodies found in memory B cells of 3 infected individuals. And although the RBD accounts for most of these antibodies, in agreement with what we observed with serum antibodies, we see that the N-terminal domain accounts for 5 to 20%. There's variation across individuals of monoclonal antibodies cloned from memory B cells. The best of these antibodies, shown in color, are actually very, very potent. And they also neutralize SARS-CoV-2 with single-digit nanogram per mole midpoint neutralization. And that's very, very encouraging. And analyzing of all of these antibodies led us to delineate multiple antigenic sites through binding assays. And Matt McCallum, a postdoc in the lab, actually structurally delineated these antigenic sites as shown here with different colors for the different antibodies that are prototypical members of these antigenic sites. So the NTD accounts for some neutralizing, the NTD accounts for antibodies of various types, targeting it in different ways. And we here again define an antigenic map to show which are the sites that are targeted by antibodies. The question was, which ones are important? Which ones are neutralizing? Which ones are not? It turns out that among the 40-ish antibodies we looked at from the 3 individuals that are targeting the NTD, only antibodies targeting the antigenic supersite or site 1 that is shown through this red antibody were actually neutralizing in vitro. All the other antibodies were associated with just binding, but no neutralization. And at the same time as our work was done, other groups came to the same conclusion. There is really one site of vulnerability on the N-terminal domain. The question is, how important is it for in vivo protection? We saw that the antibodies in vitro are just as good as the best RBD antibodies. So again, in collaboration with Johan Nates, we characterized in vivo protection using prophylaxis and Syrian hamster. And we observed very good protection at 2 doses, 1 mg per kg, which was the same as what we observed with RBD antibodies, or 4 mg per kg, which is slightly higher. However, I'm sure you noticed that we have a few outliers here. Viral replication was shut down in most animals, but not quite all of them. So we asked, what's going on in these animals? Why is it different from what we saw for all RBD antibodies thus far in the exact same challenge model? So by sequencing virus isolated from the lung of these challenged animals, we saw that from each group, we sequenced 1 representative animal, and the virus resulting from the sequencing efforts had a deletion at residue 144. The deletions are larger, but are encompassing residue 144, and that's present in both animals that we sequenced. This deletion was not present in the input virus or in the control group that did not receive a SARS-CoV-2 antibody. So these data really suggest that the selective pressure resulting from antibody administration led to the emergence of escape mutants, because this position 144 is located right in the middle of the antigenic supersite which we identified. And that really highlights that the NTD, although being targeted by very potent neutralizing antibodies that are protective in vivo, is mutating really fast. I'm showing you on this plot the prevalence of mutants in SARS-CoV-2 isolates that have been sequenced to date. There are more than 500,000 genomes that have been reported, and the height of the bars represent the prevalence of each mutant, and you can see that the NTD is mutating like crazy, a lot faster than even the RBD, which is also targeted by neutralizing antibodies. Actually, what Matt was able to show using a very large library of NTD mutants that he built, and that binding of most of these potent neutralizing antibodies was abrogated by many of these mutations. So white means no effect, red means binding is abrogated. And you can see that position 144, for example, is actually abrogating binding of almost all the neutralizing antibodies, except one. S2L20 is a non-neutralizing antibody, which is hardly affected by any of these mutations, really supporting the idea that there is a selective pressure applied by NTD antibodies that is leading to the emergence of these mutations. What's really interesting is that the Y144 deletion is actually present in the B117, as known as UK variants. And we actually showed that most neutralizing antibodies targeting the NTD are actually losing by a factor of 10, 100, or 1,000 times the neutralizing potency toward these variants. And many other groups, including David Ho's group at Columbia, made similar observations. So I would like to point out that here we were looking at the effect of single mutations, but here we looked at the effect of the constellation of mutations present in the NTD of the B1351, as known as South African variant. And you can see that the constellation of all mutations leads to loss of binding by all these antibodies, which is very concerning, and again, supporting the fact that antibody-mediated selective pressure leads to the emergence of escape mutants. So one of the reasons for the NTD to be mutating so quickly might be due to the fact that it comprises a single antigenic site that is important for neutralization, but that's still a working hypothesis. In the last few minutes, I would like to tell you how this work really guided our effort to design the SARS-CoV-2 vaccine. And at this point, I hope I convinced you that the RBD is very important for neutralization and is the main target of neutralizing antibodies in COVID-19 patients. This is the plot I showed you from our work, and Jesse Bloom's lab, as well as many other labs, subsequently made similar observations that when you deplete RBD antibodies, you deplete neutralizing activity by 90–95% or more. So the RBD seems like an ideal target for a vaccine. The problem is that the RBD is a small domain, 20–25 kilodalton domain, and that is therefore not very immunogenic. So how to go about that? And so that leads me to the fantastic collaboration that we established with Neil King's lab in our department to use the technology that he had developed to multivalently display viral antigens to make vaccine eliciting high titers of neutralizing antibodies. So very soon after the first SARS-CoV-2 isolate sequence was released, Lexi Walls in my lab and Neil King's lab teamed up to be able to tackle this question. So the approach we decided to follow was to graft the RBD through genetic fusion to a computationally modified trimeric protein that was designed in Neil King's lab that was known to be able to self-assemble with a pentameric protein that also came from Neil's lab to form this self-assembling nanoparticle displaying 60 copies of the SARS-CoV-2 RBDs in a highly immunogenic array. We decided to compare this vaccine candidate in a preclinical study to the SARS-CoV spike, which is used by SARS-CoV-2 spike, which is used by most vaccines that we know of, and just the free RBD as a benchmark. So these beautiful nanoparticles are actually assembling as seen by negative stain EM and retain the antigenicity, meaning that the RBD displayed at the surface of these nanoparticles is reacting with monoclonal antibodies and ACE2 as is expected for the RBD, which is part of the spike. Mice immunization showed that various flavors of these nanoparticles using different linker length or valency led to very high titers of neutralizing antibodies. The higher we are on this plot, the higher the titers of neutralizing antibodies. And what was really fascinating, this is the mouse study, I do not remember if I mentioned that, but the neutralizing antibody titers elicited by the nanoparticle were much, much higher than the prefusion stabilized 2-piece spike, even at a 5 times lower dose. We compare here at best to the titers shown here. The nanoparticle was also eliciting much higher titers of neutralizing antibodies than what is found in human convalescent sera, the COVID-19 patients. In collaboration with Tim Sheehan and Ralph Berick's labs at UNC, we were able to show that the vaccine was actually protective against SARS-CoV-2 replication, both in the lung. Here we are looking at the virus flavor of the nanoparticles, sorry about that, as well as in nasal turbinates. So through a collaboration with Deb Fuller, Lexi was able to analyze antibody responses in pigtail macaques and show that the vaccine was actually elicited a broad diversity of antibodies against the RBD. And I'm showing you 3 sites with 3 colors, and we see that polyclonal antibodies elicited by the vaccine competed for binding to the receptor-binding motif, as shown in blue, the CR3022, which is antigenic site 2, or the 309 antibody, which I mentioned earlier in my talk, which we call site 4. So it's very interesting because the diversity of antibodies elicited is probably going to be very important to cope with the emergence of variants due to the widespread, the global spread of SARS-CoV-2. So finally, through a very large study supported by the Gates Foundation in collaboration with the King Lab and the Pudendran Lab, we were able to evaluate the efficacy of this vaccine in a large non-human primate study. And various clinical adjuvants used with the vaccine led to the elicitation of very high neutralizing antibody titers, except for one of them. And so we were very happy to see that. And the vaccine was actually protective against viral replication with all these adjuvants, except the one that was not doing very well here, in the higher and the lower respiratory tract, which is very, very encouraging. So we are looking at control group. Here, the higher, the more viral replication we have. And we see that, for example, ASO3, which is the lead clinical adjuvant, is completely, in conjunction with the vaccine, is completely shutting down viral replication in the upper and lower respiratory tract, except for a little bit of replication for one animal. So that's actually very, very encouraging. And it was possible to see that there was actually no sign of disease in the lung of vaccinated animals with 2 different adjuvants as compared to unvaccinated animals. So this vaccine actually entered a phase 1/2 clinical trial at the end of January. And we are really, really excited to be able to see how well it will perform in humans. So I think my time is up. So I would like to conclude by thanking the fantastic members of the lab who did the work. And I mentioned some of them along the way, but really this is a team effort. It takes a village to achieve that. So I'm really, really grateful for all the work they have done throughout the pandemic. And also our fantastic collaborators. I mentioned many of them throughout the presentation, but I would like to highlight again the King's Lab at the Institute of Protein Design, with whom we work in close collaboration for vaccine design, and the Fuller's Lab, who also provided instrumental contribution to this project, and David Veesler's lab at Vir Biotechnology, with whom we did the antibody discovery and characterization work. So with that, I would like to thank you for your attention, and I would be happy to take any questions.
Yeah, David, thanks for putting the members of the lab up there. Sometimes when we're actually able to do this in person, I'm able to invite them to stand for some recognition, but let me applaud them, at least one person truly, and I'm sure everybody tuned in appreciates the outstanding work that this group has done. I'm looking at the Q&A right now. I don't see questions coming in right now. Maybe those of you who are scratching your heads or chins a little bit would go ahead and type in either the Q&A or the chat any questions. One just came in here. What kind of animals were used in this work?
So because I'm not sure which specific part, I will try to answer the different parts. So the mouse work is bulb C mice. The first part with the polyclonal antibody competition I mentioned was actually coming from a pigtail macaques from Deb Fuller's group. And the last part, the large NHP challenge study, came from rhesus macaques.
So many coming in right now. Any idea what the mechanism of neutralization is for the NTD targeting antibodies?
Sorry, can you repeat the first question? Any idea what the mechanism of neutralization? Well, that's something, yes,
yeah.
Something we're trying to figure out. There is a variety of alternate receptors or attachment receptors that have been proposed in the literature, and we are trying to investigate them. Although the ACE2 effect on entry is really clear-cut, it is not very easy to reproduce some of the data in the literature in terms of alternate attachment receptors. We suspect that they may work through blocking interaction with an alternate attachment receptor, but we have not been able to show that yet.
Okay. Another question here, how do we compare your studies with injection of proteins as antigens to the AAV vaccines and the mRNA vaccines? He's arguing it would seem like a comparison of apples to oranges.
So, I mean, that's something we would love to do. I mean, this is something that Neil and I talk about all the time. We would love to be able to compare side by side vaccines that are in the clinic or actually deployed in the population. So I guess the comparison would have to be part of the same study, because one of the difficulty we have found, there are multiple aspects. I'm not sure what the question is referring to, but if it is an in vitro neutralization, then that will have to be done by the same assay in the same lab. If it's in vivo protection, here again, there are variation across studies, including the viral titers used for challenge, which makes it almost impossible to compare studies. People are detecting barriers using different primers. Some people are challenging with 10 to the 5, some with 10 to the 6 PFUs. So the only way to do that would really be to have a side-by-side benchmarking of these different vaccines. And the only parameter that would be left is the dose administered, but presumably we could compare with the dose used in the clinic or matched to the weight of the animal. I hope, Mike, that's beginning of an answer to your question.
Yeah, do you see a follow-up question there from Mike? What about the fusion machinery of the spike as an antigen?
Well, so that's something very interesting and that we've been thinking about. Actually, Jason McClellan's group has been doing a little bit more than thinking about it. It recently showed that they had implemented that for the MERS-CoV spike, and they were able to elicit some protection in mice. Although I do not know what the PFU titers were for challenging. And here again, I'll be curious to know more details. It was one slide in a presentation, but that's an idea that is very good. I think the first step would be to learn about the titers of fusion machinery antibodies in infected individuals, their potency. The data that we and others have so far suggest they are not as potent, so we will have to compensate by eliciting higher titers of them.
So here's a question. I mean, the data that you showed on the multi-mutant kind of spike protein and the lack of the interaction, the neutralization with the antibodies was a little disconcerting. Question here from Ethan Merritt. Do you have any plans or preliminary results from your designed multivalent vaccine constructs that would, I guess, present or bind to the multivariant RBDs?
We have plans, not preliminary results yet, but let me just say that some of the variants of concern that have emerged have affected the vaccines that are currently deployed in the population. The mRNA vaccine from Pfizer and Moderna, they led to a decrease in neutralization potency in vitro by about 6 to 8-fold. And there was a concern by some people that the RBD, because it's a smaller antigen, will be more sensitive to these mutations. So we have compared side by side in our assay, and actually we see the same decrease. So there is no disadvantage in that specific regard of an RBD only based vaccine, but just like the other companies, we have to reformulate our vaccine to account for that. So we have plans, but no data yet.
Okay, another one here from Kyle. Have you found any neutralizing antibodies targeting the conserved S2 fusion machinery? And why is the S1 subunit immunodominant?
So we have actually started a mouse study 4 years ago, and with the goal of identifying fusion machinery targeted antibodies. And that's work that I did not have a chance to show, but that was led by Max Sauer in the lab. And he has found an antibody targeting a conserved epitope, and this antibody cross-reacts with more than 5 different human spike glycoproteins and neutralizes spike from 2 different lineages, although not SARS-CoV-2. So there are antibodies targeting the fusion machinery. But so far, what we and others have observed is that they are not as potent. So we will have to figure out how to elicit much higher titers of them to compensate for that in the vaccine. The other question, part of the question was about the S1 subunit. So there's clearly a difference of immunogenicity between the apex and the fusion machinery. We thought that it could be due to accessibility, but recent tomographic work on SARS-CoV-2 showed that spikes are actually quite sparse at the surface of the virus. So it's probably not a problem of accessibility. And so it's probably the intrinsic nature of the structure and sequence. There are a bunch of glycans everywhere, so I'm not really sure yet why the S1 subunit is more immunogenic, but there is no doubt it is.
So David, you might see these questions in a bit of a follow-up from Alex Mers. Jeremy Camel and Ben Hurley looked at delta Y144. In our patient cohorts, it is more strongly neutralized. Might this have to do with the fact that Y144 falls within the sugar binding domain?
So there's no known sugar binding domain in SARS-CoV-2, but this site is not far from the sugar binding domain in MERS-CoV, for example, or OC43. I have not seen Ben Hurley's data, but that's in discrepancy with all the literature I'm aware of, because this is the mutation that is present in the N-terminal domain of the B117, as known as UK variant, and that leads to a complete abrogation of neutralization by NTD antibodies, almost complete, not only by our group, but by this is what other groups have seen. So I need to look and see what could explain that difference. I'm not sure.
Okay, from Ellen Wiseman, do your results inform studies of bat viruses that might be future problems? People thinking, to your knowledge, along those lines? Think they are.
One of the things we are thinking about is, instead of making a vaccine that will work only for SARS-CoV-2 and a limited set of variants, is to identify the most conserved epitopes, not necessarily in the fusion machinery. We have shown cross-reactivity between SARS-CoV and SARS-CoV-2 is possible. So the SARS-CoV vaccine does not necessarily require to use the whole spike. But so identifying these epitopes, understanding which ones are conserved, which ones are not, which ones are protective and which ones are not, will eventually guide the design of vaccine that will elicit broad protection, at least within a viral subgenus. And the goal will be to be much broader than that in the future.
So do you see this one from Jeremy Kamal? We have looked at Del 144 spike with Ben or Lee. It looks more sensitive to patient antibodies using VSV spike virus. So Del 144 seems to be more easily neutralized. I thought maybe this has to do with effects on the sugar binding domain. Since this is opposite what you see, I wonder if you have any thoughts.
Again, there's no sugar binding domain that is known really in SARS-CoV-2. I mean, the heparin sulfate or heparin has been proposed to bind to almost every domain of the spike. But again, some of these assays we are not able to reproduce in house. So I'm actually interested to understanding more about that difference, because again, that's not really on par with what we or other people have observed in the literature.
Okay, good. Let's see. An anonymous attendee, can you speak to the possibility of vaccine-induced escape variants to SARS-CoV-2?
Well, I think we're going to have to wait and see, but I mean, I think one of the leading working hypotheses is that because protection against the B117 UK variants seems to be pretty good for all the vaccines, wiping this one out with vaccines, or at least decreasing significantly transmission, should be possible. But we know that the 2 other variants of concern, the B1351 from South Africa, as well as the P1 from Brazil, has emerged in regions where the attack rate was very, very high, and the convergent mutation that have emerged in response to most likely antibody responses is concerning and might be a problem for people that have been vaccinated with the first wave of vaccines. So it is possible that these variants could become dominant once everyone is vaccinated, because they escape partly neutralization.
So
That's
Mike sorry, has a question here about the mass production of the nanoparticle vaccine versus AAV and the mRNA vaccines. Neil, I've heard say says that the components, the ingredients required for forming the nanoparticle vaccines, I believe, are pretty readily available and in good supply. So
yeah, it scaled up very, very well, and it's stable at room temp. So we are hopeful that that can make a difference as well, because it's a very different mechanism of storage than what's is currently on the market.
we have a listener here who is asking whether or not it's possible for her to volunteer for the nanoparticle trial. I think those are underway in Korea, so it might be a little tough.
I would have volunteered myself, but I think it's difficult. It would require going to Korea, and I'm not sure they would let us come in at the moment, coming from the US. So I don't think so.
So hopefully we'll get some going in the US. And from Jose, why has the RBD not elicited mutation to select for non-neutralizing antibodies?
Sorry, let me
you see that one? Why has
the RBD not elicited mutation?
Not sure I understand.
Sorry, yes. So there are mutations in the RBD that have emerged and that are actually also detrimental to neutralization. So most neutralizing activity targets the RBD, and some mutations, the same mutations that independently emerged in multiple viral lineages, and they lead to decrease of neutralization. So currently, what we see is that if you use an RBD-based vaccine or spike-based vaccine, most neutralizing activity is toward the RBD, as is the case upon natural infection. NTD antibodies are definitely elicited by spike-based vaccines, not our vaccine because the NTD is not there, but it appears that the ability for the virus to escape NTD-mediated neutralization is very, very high due to mutations. And therefore, it's still unclear, the question is still out there, how important these antibodies are for in vivo protection. And we just don't have the answer yet.
Great. David, we've got 1.5 minutes, I think. Outstanding talk. Thank you very much. Obviously, an awful lot of interest and questions. And let me say once again to your research group that you folks all and the collaborating laboratories have done truly outstanding work, and it's much appreciated by an awful lot of folks. Thank you very much.
Thank you very much.
Good. Bye-bye.
Bye-bye.